Hello everyone, Namaskar, warm welcome to all of you. This is Chapter 13, Photosynthesis, Part 1. In this chapter, we have to discuss basic features of photosynthesis. How can you define photosynthesis and what is the significance of photosynthesis? Then we have to discuss some early experiment, that is historical account of photosynthesis, which laid foundation of this photosynthesis. And in the last of this part, we have to see a site of photosynthesis, that is chloroplast. This is Chapter 13, Part 1. This is Chapter 13, Photosynthesis in Higher Plants, and Part 1 of this. You know, children, this chapter is a basic chapter. In your earlier classes, you know what we mean by photosynthesis. You have gone through. Now we have to see detail of this. Photosynthesis, you know, we can define it. It is a physico-chemical process. What happens in this process? That this is the green part of the plant. Let this one is the leaf, green part of the plant. What it will do in presence of sunlight, it will take carbon dioxide of the atmosphere. Chlorophyll is inside the leaf itself, that is, this is green, and water is here. In presence of all these four factors, the two factors here are external, and two factors, that is chlorophyll and water, these are found inside the plant. These green plant, they will prepare the food. They will trap this light energy, and as a result of this part will make the food that will synthesize organic substances. And mainly we can take these organic substances, mainly this one is, that is carbohydrate. This process is known as photosynthesis. Is it clear? Can you summarize it? This is a physico-chemical process. Green part of the plants means that which have chlorophyll. Green parts of the plants, what they will do? They synthesize or manufacture complex organic substances in which carbohydrate is one main thing. But remember, other substances are also synthesized which are organic in nature. And for this synthesis, what they want? They need carbon dioxide and water in presence of sunlight. As a result of this overall process, that is, the oxygen will be released here. Oxygen is released as byproduct. So in this process, we can say that is energy of the sun is converted into chemical energy. This is anabolic process. Why anabolic process? That some synthesis is there, you know. Synthesis is there. This is endergonic reaction, requires input of energy, absorbs energy. And this is oxidoreduction process. What we mean by, I have to tell you in detail of this. How can you define photosynthesis now? That is, it converts radiant energy to form, that is energy that can be used by biological system. A simple equation which can represent photosynthesis we can write, that is, this is 6 CO2 12 H2O in presence of light and by green part of the plant, that is carbohydrate will be produced, synthesized, and oxygen is released as byproduct. You see here, important one is, very important, this one is, what we mean by oxidoreduction process. Remember here, in this overall process, the carbon dioxide that is reduced, remember this is reduced, and the water is being oxidized. So here we can say that is reduction and oxidation that take place together. So photosynthesis overall is called, that is, oxidoreduction process. Is it right? Come to the next now. What is importance of photosynthesis? Even a layman knows, if there is no sun, means there is no energy. It will be rather impossible to survive on our planet. There will be no life. Means sun energy or sunshine, we can say that is sunshine is important for, that is, life. What we mean by this, in sun or in that is by utilizing sun energy, the plant do the process of photosynthesis, and by doing so they prepare the food, and this food will be used by the plant itself, and the remaining that will be used by the whole universe, by all that is livings, that is they will use this food, almost all livings. A few exceptions are there, that is. So how can we say what is the significance, what is the importance of photosynthesis? That this is the primary source of food on earth, also responsible for release of oxygen. In this process I told you, oxygen is released. This will be released in air, and what will be now that this oxygen, that is required by all life forms which are doing aerobic, that is respiration. The third important point, oxygen released in photosynthesis is responsible for making ozone layer, ozone filter which surrounds our earth planet. This is our earth planet, and earth planet, this is surrounded by, that is, this is the ozone layer. This ozone layer was formed for the first time by the organisms which were there in water on earth, and they did photosynthesis. And these organisms, these were blue-green algae which are there in water. They did this oxygenetic photosynthesis. Important term is that is, they did oxygenetic photosynthesis. Photosynthesis may be non-oxygenetic also, which is done by bacteria, but here we are concerned with higher plants. So this blue-green algae, they did oxygenetic photosynthesis. They release oxygen in atmosphere, and this oxygen combined with nascent oxygen, it has made this ozone layer. Because of formation of ozone layer, all these harmful cosmic rays have been filtered, and the life from water, it has come on surface of earth. Very, very important point you have seen by now, that is significance of photosynthesis. Now we have to see some historical experiments. That is, before that we have to see that 4 factors I told you are necessary for photosynthesis. How can you prove that this light and chlorophyll is necessary for photosynthesis? If a plant organ has done photosynthesis, the basic thing, it means it has prepared the starch. By testing this starch, if the starch test is positive, means starch is there, means photosynthesis is done. By doing this starch test, starch test is done by dipping in iodine, and if that turns, that is blue-violet, it means starch is there. It means that part has done photosynthesis. You have to remember this basic thing here. Want to prove that light and chlorophyll is essential. Very important point here is, first we have to start our experiment. Before we start, the plant is to be de-starched. De-starch means starch is already present in this. We have to consume this starch. For this we have to keep the plant in dark at least for 48 hours. Result will be here. This plant will be without starch now. And now we have to take this variegated leaves. You know the meaning of variegated leaves? The leaves, that is somewhere it is having this, that is colored spots. The chlorophyll is present here, and this is the place which is yellow, which doesn't have chlorophyll. Cover it 2 to 3 leaves with black paper. Expose the potted plant sunlight for 1 to 2 hours. This is also important. That is longer time we have not to put in sunlight. Pluck the leaves, one covered and one exposed, and test them for starch. Simple thing. That is, if the leaf is covered with black paper, that will not make the photosynthesis, and here the starch test will be -1. You can see here that this is the black paper strip we have kept here even on the same leaf. That is here the photosynthesis will not be done, and that is the part where we do not have starch. This part is exposed, that is this is the area unaffected, that is with iodine. Is it right, this one? And this is the part which shows positive test. This is the part which shows negative test. We can say that light is essential for photosynthesis. Is it clear? The same thing we can do here, that is this is variegated leaves, that is the part of the leaf which is green, that will make the starch, will give positive test, and where that this starch is absent means that was the part where chlorophyll was absent. Is it clear? Now we have to demonstrate that carbon dioxide is essential for photosynthesis. For this we have to go that a popular experiment is there called Mohs half-leaf experiment. This is in the same way. This is a 𝕆𝕋 plant. This is essential 𝕆𝕋 plant. This one is. We will take this 𝕆𝕋 plant. This half-leaf is inserted inside the bottle consisting of this potassium hydroxide. Half is outside, and we will put this again from 1 to 2 hours in light. This question came 2-3 times. What's the significance? We cannot put the plant for a long time here, and we will go for a starch test. The part of the leaf which is outside, that will give positive test, and the part of the leaf which is inside, that will give negative test. You see, children, here, that is we cannot put this plant in light for a longer time. Reason is, if it will be put for a longer time, this is the part where the starch is formed. The starch can move from this part to the part where the starch is absent. Therefore, we have not to put that for a longer time. Now we have to say historical accounts, that is early experiments. Very classical experiment is of, that is Priestley experiment. Priestley, that is he discovered oxygen, that is he observed that a candle burning in a closed space, a bell jar, it will extinguish soon. Similarly, if a mouse is kept herein, then this mouse will also, that is, die because of suffocation. But when he placed a mint plant means that is having chlorophyll, that is in the same bell jar, he found that mouse stay alive and the candle continue to burn. Priestley hypothesized that plant restore, that is by burning candle or by putting, that is mouse, the air is being damaged here, that air is being damaged, and this damaged air, that is, can be repaired by this plant. Now you can summarize that the plant will release, that is this plant that will, that is release oxygen and will take, that is carbon dioxide, the carbon dioxide which is being added by burning of candle, or that is by this live mouse. Is it clear? In Ingenhousz, he performed very classical experiment. Similar setup what were used by Priestley, by placing it once in dark, important. The same experiment, if it is placed in dark, then the green plant was not able to purify the air. He proved that the sunlight is essential for the process. And then he, in an elegant experiment, he proved that in this process, that is oxygen is being released. For this, he took this aquatic plant. This is an aquatic plant, and this aquatic plant that was exposed to sunlight, here it will do the process of photosynthesis, as a result of which bubbles will come out, which later on have been identified as oxygen. Von Zech, in 1854, he provided evidences for production of glucose when plant grow. Glucose is usually stored in the form of starch, and his studies showed that green substances are essential, that is, the chlorophyll is essential, and now this green substance is identified as chlorophyll, which is located in the body chloroplast. Means sac, he told glucose is formed. he told chlorophyll is there, and chlorophyll is present in, that is, chloroplast. Is it? A very, very classical experiment was done by 𝕆𝕆𝔻𝔼𝕆. That is, the question related to this, a first action spectrum of photosynthesis was given by Engelmann when much more detail was not known that time, he proved, that is, in which color of light, that is, in which wavelength of light maximum photosynthesis is there. He split light with the help of a prism. This is the prism. The light, it was split into, that is, rainbow colors, Vibgyor, and then one by one, that is, this was being exposed. This was given to this green alga, that is Cladophora. Cladophora is a filamentous algae, and it was kept, that is, in a suspension in a water where the bacteria, that is, were kept. These were the aerobic bacteria. And you see the experiment. That is, here this is the Cladophora, this is the tube, and here we have, that is, these are the bacteria present, aerobic bacteria. When the rainbow spectrum was given, it was found that the bacteria, they got accumulated here in, that is, blue region, and then in, that is, red region. Means in blue region, that is, maximum oxygen was being released. Here also oxygen was released as compared to other regions, and therefore these aerobic bacteria, they got accumulated here. So he gave an idea that blue light, or this part of the spectrum, blue and red, that these are more effective as compared to the other part of the spectrum. Is it clear? Then come to the next now, that is, here by middle of 19th century, very important statement, the key features of the plant photosynthesis were known, that the plant could use light energy to make carbohydrate from carbon dioxide and water, and empirical equation was represented, that the total process can be represented like this. This is carbon dioxide, this is water in presence of light, and in presence of, that is, chlorophyll, that will be formed, that is, and oxygen will be released. It will be represent the, that is, carbohydrate, glucose, a 6-carbon sugar. Very milestone contribution to understand photosynthesis that was given by, that is, von Neil, who was a microbiologist. Milestone contribution, remember what he told. You see, Neil, being microbiologist, studies was based on purple and green, that is, sulfur bacteria. What he told, he demonstrated that these are doing photosynthesis, but they do not use water as source of hydrogen. Instead of water, they are using, that is, hydrogen sulfide, remember. So he told this thing, that is, here, this is H2A. We can write that this is H2S. So this is the reduction power which will be provided, that is, which will reduce the carbon dioxide, and as a result of which, that this 2A, or we can say, that is, sulfur, will get precipitated. So he gave an idea that for reduction of carbon dioxide, a reducing substance is there. In this case, that is hydrogen sulfide. He assumed, that is, in green plant, instead of this hydrogen sulfide, water is the hydrogen donor and is oxidized to make oxygen. Similarly, here instead of 2A, oxygen will be released, and here that he told oxidation product is sulfur, as in, that is, bacteria or sulfate, that is, and not oxygen. In case of higher plant, the byproduct will be oxygen. Very classical experiment it had been, and a solid proof that oxygen released in photosynthesis that comes from water, that was given by Rubin, Kaman and Hashid. They use radioactive techniques, that when this water was labeled with oxygen, it was found that the released oxygen is also that is radioactive one. So it proved that the oxygen released in photosynthesis, it comes from that is water, and the oxygen of the carbon dioxide that goes to that is this carbohydrate. It goes to carbohydrate. Is it right? Now we have to say that is chloroplast. You know, site of the photosynthesis is chloroplast. You are familiar with. Let we are taking example here. That this is that is a leaf here. You know, if I made this dorsoventral leaf, this is the midvein here. If we cut a section of this, you know, this is upper epidermis, and this one is that is, you know, that one is lower epidermis. Here, the tissue in between two, that is known as, you know, that is called mesophyll. Mesophyll is differentiated here. That is upper layer, which are columnar cells without an intercellular space. You know that name of this. These are known as that is palisade tissue, and here this one is spongy tissue. So dear student, you know that here in mesophyll cells, number of the chloroplasts are much more. These are the chloroplasts. These are the chloroplasts. The site of the photosynthesis is chloroplast. The light which will fall on leaf ultimately will enter into the leaf, ultimately go to the mesophyll cell, and now it will enter into the that is chloroplast. Is it clear? Before we see further detail, we have to see that is, see, I told you photosynthesis does not take place in green leaves of plant, but it does so also in other green part of the plant. This is important. Green leaf is there. Actually, what is important for photosynthesis? That is chlorophyll is essential. Chlorophyll that is there in chloroplast, that is in green part of the plant. It may be leaf, mostly this is leaf, but it may be some young stem which are green. Very important point this one is using chloroplast, that is usually the chloroplast alignment we have to see. They used to align. This is gift of nature. Nothing will go waste. What is there? See, if this is a mesophyll cell, that these are the, that is meso, yes, this is mesophyll cell. What is there? If high light intensity is there, then that can harm the chloroplast. Therefore, the chloroplast will be arranged parallel to the, that is light rays here, and they will do the photosynthesis, because for photosynthesis much more light is not required. Only about 10% of the light is sufficient, which is incident on leaves. So here in highlight, that is chloroplast to be aligned parallel to this, the movement is known as that is parastrophe. In a low light, already the intensity of light is low, that is they will be arranged that is perpendicular to the light rays. Such movement is known as apostrophe. And when the moderate light is there, the chloroplast, these are the chloroplast, these are found scattered in mesophyll cell so that they can have maximum benefit. Is it right? Now we have to see structure of chloroplast. Already in unit cell also you have seen chloroplast, you know, there is membranous system consisting of grana and stroma. I can recall it now. This is the largest, that is cell organelle, which consists of two membranes, outer membrane and the that is inner membrane. This is the outer membrane, which is, this one is the that is inner membrane. And here you see that here we have the membrane, third system of the membrane we have here, that is the system of membrane. It is called that is granum, and granum, the top granum is known as that is called, it is called 𝕆𝕋𝔼ℎ, and there may be so many grana in a chloroplast, usually in a typical chloroplast, and all these grana, these are connected by that is this intergranular lamellae. So this part is called, you know, it is called granum, and you know granum is the site of light reaction. That's why granum is having that is chlorophyll, which will trap the light energy. The remaining part, that is called stroma, or also called matrix, and matrix is the site of dark reaction. You can go through. That is, there is a clear division of labor in chloroplast. The membrane system is responsible for trapping light energy, I told you, and when they will trap the light energy in granum, that is, energy will be trapped and stored in the form of ATP and in the form of, that is, NADPH. This is stroma, that is, here enzymatic reaction will be there, where actually either C3 or that is C4 cycle will take place, and as a result of which the food is prepared. The former set of reactions, since they are directly light driven, they will be known as photochemical reaction, and as a result of this, that is, they have produced ATP and NADPH2. The latter are not directly, this one dark reaction that doesn't need light directly, it uses the assimilatory power produced in, that is, light reaction. Hence, that is, to distinguish the latter, they are called by convention dark reaction. But this dark reaction, this is a misleading word. It is misnomer. Dark reaction doesn't mean that will take place in dark. The only thing here is that doesn't need light directly. Therefore, it is, that is, independent to light. It is independent to is light. We have to remember it. The same thing we can see in this diagram. That is, this diagram, as I told you, outer membrane, inner membrane. These are the stroma lamellae. This one is the grana. That is, this one is the stroma. In the stroma, you know, ribosomes is there, which is 70S style. These are the starch granules. Beside starch, some other substances are also produced, that is, lipid. Therefore, in stroma, we have enzymes for, that is, carbohydrate synthesis, for, that is, protein synthesis, and for, that is, fat synthesis. This type of enzymes are produced. Even because, that is, their DNA is present, therefore, DNA replication enzyme are also present here. Already I told you DNA replication enzymes. And this is the part you can differentiate here. This is light reaction. This is dark reaction. That is in granum. That is stroma. This requires light and chlorophyll, and as a result of which here the oxygen will be released, and here, that is, this power produced ATP and NADPH2 will be used here. Remember it. This is part 1, chapter 13. Thank you.